are TUBES reagents, which are high-affinity ubiquitin binding reagents for purifying and detecting polyubiquitinated proteins. TUBES have really changed the way that ubiquitin researchers can characterize and study ubiquitinated proteins. And it's really opened up the door to being able to study endogenously ubiquitinated proteins in any sort of experimental system that the researcher desires. And prior to the development of TUBES, characterization and isolation and purification of ubiquitinated proteins was limited to systems where you could transfect in tagged forms of ubiquitin that you could use as a handle for purifying these proteins. And now with the advent of TUBES, this opens up the opportunity to do these purifications from cell lines that are not accessible to transfection or tissues or other normal or pathological situations where transfection is just not possible. So before I begin and start talking about TUBES, I'd just like to also introduce LifeSensors briefly. We're an established company founded in 1996. We're located in the east coast of the United States, just 35 miles west of Philadelphia. And over the years, we've built up a catalog surrounding two sort of technology platforms. The one shown here on the left is a technology platform that facilitates high-yield protein expression using fusions of proteins of interest to the ubiquitin-like modifier SUMO. And this has been shown to improve the yield of not only total protein, but also soluble and functional protein. And so if you're interested in improving the yield or the efficiency of your protein expression, I would encourage you to go to the LifeSensors website or contact your TUBE representative or LifeSensors directly, and we'd be happy to introduce you to our protein expression platform. But what we're here to talk about today is our LifeSensors offering of ubiquitin pathway reagents. We have about 250 products that are useful for interrogating the ubiquitin proteasome pathway, and these include ubiquitin affinity reagents like the TUBES that we're going to be talking about today, as well as a strong portfolio of ubiquitin pathway enzymes and assays for measuring activity and doing drug screening, specifically with the ubiquitin pathway. We sell a number of ubiquitin derivatives and chains. We also offer ubiquitin microarray services, which are useful for doing screens to identify enzyme substrate pairs within the ubiquitin pathway. And lastly, we have a portfolio of small molecule inhibitors that are useful for doing cell biology experiments and inhibiting various aspects of the pathway. So a few basics about the ubiquitin pathway for those of you who are relatively new to studying ubiquitin. Ubiquitin is a 76 amino acid protein that's conserved across eukaryotes. It's actually one of the most conserved proteins within eukaryotes. And it's the founding member of a family of ubiquitin-like proteins that includes members that may be familiar to many of you, including SUMO, NED8, and ISG15, as well as a number of more esoteric members like FAT10 and URM1. And these are all whole protein post-translational modifications. And through the C-terminal glycine on these proteins, they're conjugated to various substrate proteins in a number of different ways. And ubiquitin in particular can be conjugated to substrate proteins either as a monomer in ubiquitination events that are termed monoubiquitination. You can have multiple monomers placed on a single substrate. This is called multi-ubiquitination. And then because ubiquitin contains lysines in its own sequence, ubiquitin can be conjugated to itself to generate these higher order polymers of ubiquitin on substrates, and this is termed polyubiquitination. There are 7 lysines located on ubiquitin, and each of these is capable of forming polyubiquitin chains. And it's been shown that chains formed through the different linkages, both lysine 48 and lysine 63 are designated here in this structure. These chains formed through these different linkages form different higher order structures. And so an extended form here, characteristic of K63 linkage, is shown in this schematic, whereas K48 linkages forms a more compact structure. And so these different structures and these different forms of ubiquitination are known to have different functions and different functional outcomes for proteins that have these modifications. The most predominant and well characterized is, of course, K48 linked polyubiquitination, which predominantly leads to degradation of proteins through the proteasomal pathway. But we now know that ubiquitination and polyubiquitination has functional roles in almost every aspect of cell biology, and so it's a very exciting field, and there's a lot to be learned about how much of this coding potential the cell utilizes for this post-translational modification. So the next slide shows some of the enzymatic machinery that's used to conjugate ubiquitin to proteins, as well as remove ubiquitin from target substrates. So ubiquitin is added in a three-step process involving a set of enzymes characterized as E1, E2, and E3. So free ubiquitin and hydrolysis of ATP generates a ubiquitin thioester with ubiquitin E1 enzymes. These are then transferred to ubiquitin conjugating enzymes called E2 enzymes. And then the substrate recognition part of the pathway is regulated by E3 enzymes, which specifically bring substrates into the complex, where then ubiquitin can be transferred either directly from the E2 to the substrate or through a covalent intermediate with the E3. Ubiquitinated proteins then go on to have altered subcellular localizations, associations with different factors, or they get degraded by the proteasome into peptides and ubiquitin, which then are recycled back through the process. In addition, in opposition to the conjugation side of the pathway, there are about 100 deubiquitinating enzymes in humans that will remove ubiquitin from target substrates to regenerate the native substrate. So LifeSensors offers a number of tools to study the ubiquitin pathway, and this is just a sampling of a few of the unique products that we offer. And no matter what your particular approach is within your laboratory, LifeSensors offers a number of different products that can be useful for that approach. So we have here a set of products that are useful for detecting ubiquitin, both by Western blot as well as in cell-based methods. Very nice monoubiquitin, monoclonal antibody against ubiquitin called VU1, TUBES, which we'll be talking about in a moment. We have a ubiquitin ELISA that's useful for quantitating ubiquitin from and ubiquitinated proteins from cell and tissue lysates. If you're interested in purifying and isolating ubiquitin, we'll be talking about TUBES for the rest of this presentation. We have a number of products for assaying the enzyme activities of both conjugating enzymes as well as the deconjugation side of the pathway. A number of small molecule inhibitors, including a pan-DUB inhibitor called PR619, as well as inhibitors to the proteasome, inhibitors of E1 and E3 enzymes. And together with TIBU, we offer a set of silencex knockdown cell lines, which are a unique expression system for expressing RNAi against a variety of targets. There are about 100 stock cell lines that are available within a week, and then you can also take advantage of the custom knockdown cell lines that can be generated through TIBU Bio for any target that you wish. Lastly, we have some reagents for detecting ubiquitin within the cell and looking at subcellular localization of ubiquitin, as well as detection of ubiquitin within tissue sections as well. And finally, we offer these ubiquitin protein microarray services. if you have a particular substrate that's ubiquitinated, and then you would like to know what the E3 is or the DUB is that regulates the ubiquitination of that substrate, through our ubiquitin protein microarray services, we can tell you what those proteins are. So you'll notice the TUBES appears on many of these different approaches, highlighting the versatile nature of these reagents. And so for the rest of the presentation, we'll be talking about TUBES, what they are, how you can use them for your research, and what some of the particular advantages of TUBES are. So there are some problems, inherent problems with the analysis of ubiquitination, and in particular they center around the dynamic nature of this modification within the cell. And so ubiquitination of any particular substrate is in a dynamic equilibrium between the attachment and removal of ubiquitin by conjugating enzymes and deconjugating enzymes, respectively. In addition, one of the major functions of ubiquitination is proteasomal degradation. And so ubiquitinated proteins are rapidly degraded by the proteasome. And so if you were interested in capturing a particular ubiquitinated protein or quantitating it or measuring it, measuring ubiquitination under different conditions, this dynamic equilibrium and removal of ubiquitin and degradation of ubiquitinated proteins makes that process difficult. There are some solutions in that you can apply DUB or proteasome inhibitors to stabilize ubiquitinated proteins, but this comes with its own caveats and difficulties in that this can lead to toxic effects, it can lead to nonspecific effects, and so you're always left with a nagging question whether what you're seeing is truly reflective of the physiology that you want to study. As I mentioned earlier, traditionally people have used overexpression of tagged ubiquitin to isolate polyubiquitinated proteins. And this is largely because many of the antibodies generated against ubiquitin perform relatively poorly, particularly for immunoprecipitation. Using tagged forms of ubiquitin for purification purposes has some limitations in that not all systems are amenable to transfection of these constructs. And also, use of tagged ubiquitin can have a couple of problems in that you're disrupting the overall equilibrium of ubiquitin in the cell. And then also, it's been shown that certain components of the ubiquitin enzymatic pathways do not use tagged ubiquitins as optimally as they do native ubiquitin. So, keeping all of these problems in mind, Manuel Rodriguez at CIC Biogun in Spain developed these reagents that we call TUBES, which stands for Tandem Ubiquitin Binding Entities. And as mentioned in the title, these are high-affinity ubiquitin binding reagents for isolating and purification of polyubiquitinated proteins. And when you conjugate these TUBES reagents to various affinity tags or fluorophores, they become very useful for isolating ubiquitinated proteins from complex mixtures, cell lysates, tissue lysates. Biotinylated versions of TUBES are very useful for detecting ubiquitinated proteins in Western-type applications. And then we've also developed fluorophore-labeled TUBES for in situ detection. So what exactly are TUBES? These are basically tandem arrays of ubiquitin binding domains linked to an affinity tag or to augros. And so Manuel Rodriguez's group capitalized on the fact that although ubiquitin binding domains typically have micromolar affinity for ubiquitin, in nature one can find proteins which have multiple UBA domains within the same protein. And his lab surmised that perhaps this provided, through avidity effects, an increased affinity for polyubiquitin chains. And indeed, when he engineered tandem arrays of these ubiquitin binding domains, one can see increased affinity for polyubiquitin up to over 1,000-fold. and that's shown here. So the first two reagents that were developed, the first one was developed using a UBA domain from ubiquitin-1, and this is now called TUBE-1, and this drove basically a micromolar affinity for polyubiquitin down to a very low nanomolar affinity for polyubiquitin in TUBE-1. A second tube called TUBE-2 is derived from human RAD23A, and again took the affinity from about a 7 micromolar affinity for polyubiquitin down to, again, a 1,000-fold increase to a low nanomolar affinity for polyubiquitin. Importantly, these tubes have very little or no binding to other ubiquitin-like modifiers, such as SUMA1, 2, or 3, or NED8 at 200 micromolar. And this is published in a publication in EMBO Reports from 2009. In addition to showing that the TUBES reagents have high affinity for polyubiquitin, sort of a less intuitive finding was that these tubes also protect ubiquitinated proteins from degradation from both deubiquitinating enzymes as well as the proteasome. So in this top panel, basically it's just showing an incubation of a whole cell lysate blotted after a number of hours, and blotted and detected using an anti-polyubiquitin antibody. And you can see that although you can detect some polyubiquitin at the 0 time point, this rapidly disappears, presumably through degradation by deubiquitinating enzymes, because if you include inhibitors of cysteine proteases like iodoacetoamide and N-ethylamylamide, you can protect and preserve some of the polyubiquitinated proteins in those lysates. However, if you include either TUBE-1 or TUBE-2 in these incubations, you can see a much stronger protection of these proteins, these ubiquitinated proteins in these lysates. So TUBE-1 and TUBE-2, if included in lysis buffers during isolation, can very dramatically protect and preserve ubiquitinated proteins that you're trying to isolate. Likewise, tubes can protect against proteasomal degradation, and so this is a blot of p53. In the absence of 26S proteasome, you have this level of p53 in this preparation. If you add exogenous 26S proteasome, you can see you get some degradation of p53, but in the presence of TUBE-2, this degradation by the proteasome is blocked to similar levels that you can see if you block the proteasome using a small molecule inhibitor like MG132. So here we have now the TUBES reagents, which have been demonstrated to have high affinity for polyubiquitin and also are protective against degradation of polyubiquitinated proteins by both DUBs as well as proteasomal degradation. So having these reagents, what are they useful for? So by conjugating these tubes to a variety of affinity tags and fluorophores, we have been able to demonstrate that these tubes have multiple applications in isolation. So if you want, you can use these reagents for isolating polyubiquitinated proteins, for use for Western analysis, for co-IP and identifying what complexes of proteins are binding to polyubiquitinated protein. You can do further biochemistry and enzyme assays on these proteins isolated in this way. You can do crystallography, and you can also use tubes to purify the complete complement of ubiquitinated proteins within an experimental system, and characterize those proteins by proteomic methods like mass spec and other approaches. We found that these reagents are excellent substitutes for antibodies in Western blot applications, as well as cell staining. And as mentioned in the previous slide, there can be very useful tools for protecting proteins during lysate preparation. So if you aren't able to preserve the protein during isolation, you certainly won't be able to study it further in any downstream application. So the next slide just shows demonstration of the purification using tubes. So this is a HEC-293T lysate, and we using tubes here conjugated to agarose, and we're able to pull down a very substantial amount of polyubiquitinated protein from very small amounts of total cell lysate. So you're looking at pull downs from 1 and 2 micrograms of HEC-293T lysates compared to whole cell lysate of 60 micrograms of total protein. And this is just a blot detected using LifeSensors VU1 monoclonal ubiquitin antibody. So it's a very efficient reagent for purifying total ubiquitin protein. But if you're interested in looking at specific ubiquitinated proteins, this is demonstrated on this slide. Here we have 2 examples, the same type of experiment done with HeLa cells at the top, as well as 293 cells at the bottom. These are treated with TNF to stimulate ubiquitination of components of the NF-kappa B pathway. We're looking at I-kappa B in this particular experiment. And then detection by Western using anti-ubiquitin in the left panels, and then specific antibody to I-kappa B alpha in the right panels. And so you can see with the input before purification, you can detect large amounts of ubiquitin, total ubiquitin within either condition, and then you can detect specifically ubiquitinated and unubiquitinated I-kappa B alpha in using an I-kappa B alpha antibody. Purifying using tubes, you can get an enrichment of both the total ubiquitin in the sample, as well as enrichment of the ubiquitinated I-kappa B alpha in these samples, in both HeLa cells as well as 293 cells. Very little of this ubiquitin material comes through on the flow-through, so we're getting very efficient capture of much of this material. You can still see the unubiquitinated I-kappa B alpha here in this that's not being captured by the purification technique. And furthermore, you can take this bound material and do a further immunoprecipitation for whatever protein that you want. In this case, we're looking, again, at further purification using an antibody against I-kappa B alpha, and you can see a large enrichment of ubiquitinated I-kappa B alpha in the final lane here. You could then take this material and you could IP for different proteins to see, or immunoblot for different proteins to see what other proteins are bound in this complex. You could look at the quantity of ubiquitinated I-kappa B under different conditions and do a very thorough study of ubiquitination of this or any other target of interest. This is just this show is showing again another sort of approach for increasing the yield of polyubiquitinated proteins during isolation using a small molecule approach. And so we found that in the inclusion of the DUB inhibitors PR619 and 110 phenanthroline are critical for maximizing the amount of ubiquitinated protein that you isolate before doing these purification experiments. And so on the left side of this panel, we're looking at basically just whole cell lysates detected using a biotinylated form of tubes, showing total ubiquitin in these lysates in the absence of a DUB inhibitor. And so you do get relatively reasonable yields of ubiquitinated protein, but if you include the DUB inhibitor PR619, which inhibits all of the cysteine type DUBs in the cell lysate, you can get a much increased yield of ubiquitinated protein. Now it's not shown here, but you can get further enhancement with the 110 phenanthroline, which is actually a zinc chelator and inhibits the classes of DUBs that act as metalloproteinases. And so it's our strong recommendation that at any time that you're making lysates to characterize ubiquitinated proteins, to include both PR619 and 110 phenanthroline. Both inhibitors are available from LifeSensors and through TB Bio. If studying a single protein at a time is not enough for either you or your mentor, tubes are also very amenable to purifying ubiquitinated proteins for proteomic type approaches. And so this is published work from Lopez Ochoa, where they used tubes to purify total ubiquitinated protein from the breast cancer cell line MCF7, either treated or untreated with the anti-tumor drug adriamycin, and basically whole cell lysates of MCF7 cells were created in the presence of GST-tagged tubes, in this case, or GST beads alone as a control. This purification was taken through a number of additional purification steps. The material was eluted off the beads using glycine and low pH, and then that material was further purified by HPLC and run on MSMS to identify all of the proteins that were pulled down using GST tubes. And you can see here that a wide range of cellular targets were isolated and shown to be ubiquitinated using this method, providing this study with a wealth of potential targets to study the role of adriamycin treatment on MCF7 cells. So I believe over 600 proteins were isolated using this method. This slide shows application of tubes, biotinylated tubes specifically, as substitutes for antibodies for Western detection. And so this is a comparison of our biotinylated tube 2 with detection of ubiquitinated proteins from a whole cell lysate using our monoclonal antibody VU1. And so you can see with tubes we can get a much more sensitive detection of ubiquitinated proteins on this blot as compared to VU1. And importantly, this sensitivity is not dependent on boiling the blot or other denaturing steps that are commonly required to get maximal sensitivity with antibodies. And so boiling the blot is not required to see sensitive detection using tubes. And it's very easy to substitute tubes for antibodies in doing a far Western detection. Basically, it's a 1 to 1,000 dilution of tubes, just as you would with an antibody. The blocking reagents are similar, and then you simply have to change the secondary detection reagents from, say, a goat anti-mouse or goat anti-rabbit antibody to strep-avidin HRP conjugates, which are readily available from a variety of sources. They're very cost competitive with antibodies for Western analysis, costing just a few dollars per blot, very similar to what you would have with a ubiquitin antibody. This is a few examples of using tubes for cell staining. We have Tamra labeled tube 2 for the red channel, fluorescein labeled tube 2 for green fluorescence, and then a biotin labeled tubes can also be used, again, with a fluorescent or HRP conjugated strep-avidin secondary detection. On the left, these are C2C12 murine myoblast cells, and this is a comparison of detection of ubiquitinated proteins using biotinylated tubes compared to detection using an anti-ubiquitin antibody. You can see very similar patterns of staining using these two different reagents. Using directly conjugated tubes directly conjugated to fluorophores, Tamra in this case, we're showing that you can see very nice subcellular localization of ubiquitinated proteins within these murine myoblast cells. And importantly, with the directly conjugated tubes, this also has the advantage of being a single step labeling of cells rather than having to use secondary detection reagents. So everything that I've talked to you so far has been looking at tubes that have basically a pan-reactivity to all polyubiquitinated forms, including all of the different linkage types that I described on one of the introductory slides. We've also recently developed a tube reagent that's selective for the K63 linkage. So the K63 linkage, together with the K48 linkage, are two of the most common forms of polyubiquitination in the cell. And functions of K63-linked ubiquitin, in contrast to K48, are involved in processes such as DNA damage response, endosomal sorting, signaling through inflammatory pathways such as the NF-kappa B pathway, and also autophagy of aggregated proteins. And importantly, K63-linked polyubiquitin forms, it forms a structure that is an extended rather than a compact structure. And this distinction allowed Robert Cohen's group, who's now at the Colorado State University, to exploit this structure to develop a reagent that is selective for the K63 linkage. And so basically what he did was take ubiquitin binding domains that are specific for the K63 linkage, and rather than link them by a flexible linker, link them together with a rigid alpha helical linker to put these ubiquitin binding domains with a spacing and a register that was in line with the extended structure of the K63 linkage. And what this did was provided a reagent that had 1,000 to 10,000-fold selectivity for the K63 linkage over the K48 linkage. So we now offer this particular reagent in two forms: one, a biotinylated form, which is superior to K63 linkage specific antibodies for detection by Western, and then we also have a flag tagged version that's very useful for pulling down specifically K63 linked polyubiquitin. So just to show you a few examples of these applications, this is a comparison of our biotin K63 tubes with some commercially available K63 selective antibodies. So this is a comparison with three different suppliers of K63 specific antibodies. And on the left, in each case, this is detection of synthetic K48 chains and synthetic K63 chains with our biotin K63 tube 1, or with the K63 selective antibody. And so you can see in the first two cases, our reagent is much more sensitive towards detection of the K63 polyubiquitin than the antibodies. In the third case, the sensitivity was very comparable. Both our K63 tube reagent as well as the antibody were quite good at detecting the K63 linkage. But when we examined the selectivity further with this particular antibody with regard to specificity for the K11 linkage, you can see that this antibody is also recognizing K11 chains, and so it will not be possible to say conclusively that detection with this antibody is specifically detecting the K63 linkage, whereas the selectivity with our biotin K63 tube 1 is much more selective for the K63 linkage as compared to K11 and K48. So here we have a reagent that's superior than available K63 specific antibodies for detecting the K63 linkage. I'm going to skip ahead here to this slide first. This is just showing, again, the same data showing the selectivity of these reagents for the K63 linkage over K11 and K48. But this is showing this selectivity in a pull down using our flag tagged version. So again, these are SDS page of synthetically generated K63 polyubiquitin on the left, and synthetic chains of K48 linked polyubiquitin on the right, and a pull down using our K63 tube 1 in increasing amounts here. And so you can see it very efficiently, even at 10 nanomolar, will pull down significant amounts of the K63 chains, whereas at similar concentrations you pull down very little of the K48 chains, as shown here on the right. This importantly shows that as you do push up the concentration of the K63 tubes, it will begin to pull down some K48 linked chains. So you have to be very careful to stay within the 30 nanomolar to maybe 500 nanomolar range to specifically pull down K63 over the K48 linkage. This is just now extending that analysis to whole cell lysates and more real-world examples. This is CD3 stimulated Jurkat cells to stimulate K63 polyubiquitination within the inflammatory pathway. This is the input showing all of the ubiquitinated protein in these lysates, and you can see here that if you can definitely enrich for higher molecular weight K63 linked forms using our flag tagged K63 tube 1. And on the right here is showing some additional examples using TNF treated macrophages. This is untreated, and then here we're showing an increase in the K63 linked polyubiquitin in a TNF alpha treated sample. So that's most of the data. I just wanted to finish up with two more slides here to show you the various tubes that we offer. I described to you we have two sets of pan-reactive tubes, tube 1 and tube 2, which we have linked to various affinity tags for purification, so GST and HIS tags for purification using affinity resins like glutathione affinity and nickel affinity resins. We also have agarose versions of these two tubes for one-step and single-step pull-down of ubiquitinated proteins. Now, as mentioned, these tube 1 and tube 2 are pan-reactive with respect to the different linkage types of polyubiquitin. They are derived from different UBA domains, so it's possible they may have somewhat different selectivity for specific targets. But we found that in most cases they perform very similarly towards a wide range of targets. We do offer small sample sizes of each of these, so if you do want to try both to see which works best in your particular experimental system or with your particular substrate, we can make sample sizes of these available to you so that you can make a decision as to which to choose to move forward with. We do offer biotinylated versions of these and then fluorescent versions of these for detection by Western and cell staining, respectively. For the K63 selective tube 1, we also offer a biotinylated version for Western analysis and a flag version for purification. We are developing and extending this line of TUBES reagents. We are working towards developing TUBES reagents that are selected for the other linkages, including linear ubiquitin, and those reagents should be available hopefully within the next few months later on this year. And also we are developing a similar type of binding reagent to isolate polyubiquitinated proteins as well. And so if you have questions about these or are interested in beta testing these reagents as they come out, I encourage you to contact either LifeSensors directly or contact your TBU bio representatives. And so last, just to sum up, I hope I've given you a good picture of the capability of what tubes can do and shown you that these are ideal for isolating and purifying polyubiquitinated proteins. They have very high affinity for polyubiquitin chains, and this eliminates the need for expressing tagged forms of ubiquitin for purification. They're ideal for isolating proteins for proteomics, Western blot analysis, or other biochemical studies, and we've tried to link them to a number of different tags and detection reagents to meet as many experimental needs as possible.